Okay, my friends, this has turned into one of the deepest dives I've ever done, and this is about biology, and it's about the Sars Covid vaccines and proteins and spike proteins and capsids and all kinds of stuff that nobody really pays much attention to, because they started with COVID-19. They're looking at all this stuff, COVID-19, and trying to program for it. Well, let me tell you something. They have known about SARS for many, many years. This goes back to 2003, 2004. Antibody detection of Sars Covid spike and nucleocapsid proteins. Both of them they're talking about 20 years ago. Now, what is the spike protein, and what is a nucleocapsid protein, and what is a protein? What's a protein? This is the problem. People see these catchphrase words, oh, you gotta look up the spike protein, it's disastrous. Well, it does look like it could be a problem, yes. This is tricky business. This is programming, literally programming. As you will see, I'm going to get very deep into the ribosomes, which are the programs. And then how are they delivered? Are they delivered in these little capsids, which are nothing more than like seeds? They have a capsule with this program inside of it, and it has to get in to one of our cells for its program to take over the program that's in the cell. But it can only do that if it's allowed to do that. And we should have some bacteria that creates some enzyme that it will not allow it. It will say, I can see your chemical signature and you are not allowed here. Done. If you don't have the correct bacteria in your body to create the enzymes, you will not be protected, and they are in your immune system, which is just recently understood. Meet your interstitium, the newfound organ, all right, which is right here. And that is that fluid-filled highway. You go on the other side of that highway and we're in trouble. You keep them out. And the only way you can do it is with proteins that are enzymes, and those enzymes are created by bacteria. You don't have the bacteria, you're done. That's the problem. People have taken all this antibiotic stuff and toxins into their body. It's killed the living, beneficial body bacteria. And when you see this, is going to be very long, it's going to be very detailed, but it's very important. I pretty much understand it now. And I understand what COVID SARS is all about. It's a severe acute respiratory infection. And what is it? What happens in your respiratory system? You have a lot of goo. Literally, you have a lot of goo in your lungs that just keeps going, and it's a certain type of collagen, and the SARS attacks that type of collagen and converts it to another type. It turns it from the rubbery one into like jelly or something like that. I remember looking at the autopsies long ago. I have to go back and look at that. But this is going to be a ramble. It's going to be very deep, and it's going to concentrate on the ribosomes, the bacteria, these capsids, the mRNA strands. Now they found out there's all kinds of different strands. There's dual strands, single strands, there's all kinds of stuff. And again, very deep, very deep. And you see all these colors? The colors have meanings. And they're all from these transition metals, and they're called ligand attachments to little bits and pieces that carry things around in your body. And that's what a protein is. You see that? That's and it has to be exactly identical to that every time. I mean, identical. One little thing goes wrong, and it's called denatured. It's not natural anymore. It's destroyed. One tiny little click, done. And that's what we have to have, is the body chemistry coming out of there, which is these little bits and pieces that turn into these that attack the invaders. Bam! Kill them. Get ready. This is going to be deep. And a lot of doctors don't understand this. This was not even understood. This even existed. This is only 2018, a newfound organ. It's the entire body in your whole body. And this is the most important organ in your body is your interstitium. Right there, that layer right there. Most important part of your whole body. That's the fluid-filled highway where all of the immunities fight back. This is the defensive line. You break through there, and any of your kidneys, your lungs, your heart, your liver, your gallbladder, your intestines, anywhere. Lymph nodes especially. Lymph nodes are the dumps of the body, so they have to get rid of the toxins. If you don't have the right bacteria to create the right enzymes, to create the right chemistry, the toxins build up, it breaks through, and you end up with cancer or some seriously invasive disease. All right, when COVID first came out, there was a couple of autopsies done, and they determined there was a problem with the collagen. And listen to this now. This goes back to 2020, Illinois Tech, and they did some research on collagen. It says new research on collagen could influence treatment of COVID-19. Now, this is the important part right here, is in diseases affecting the lungs have a ton of rubber. I mean, literally rubber. One of the collagens is extremely rubbery and flexible and stretchy and all that stuff. That's the one that is getting attacked. So in diseases that affect the lungs, which is the rubbery parts, the viruses have been shown to cause too much collagen to be made. Too much rubber, and it just collapses the lungs. It's deposited, causing the lung pulmonary fibrosis and or autoimmunity against collagen. Well, this is where they're saying your autoimmunity, your body turns against itself. Well, here it is right here. This is where the body attacks itself with sometimes frightening complications. That right there is an assumption. The body doesn't attack itself. The body can't protect itself from this attack. That is my conclusion. All right, this is from a company that cleans water and sludge and so forth. Now listen to this. I talk about bacteria making all these enzymes. Yes, you need the enzymes, but that's not enough. You also need the bacteria. Bacteria are still needed. The actual bacteria, the little guys running around, are still needed to go out and consume the waste material. The enzymes will break the waste down, but they will not do the job of removing the waste. The bacteria has to bring it to where it has to go, and they do it by using oxidation states. As I showed you, the oxidation states of transition metals. Right here, these are different oxidation states. And that's what carries things around in your body. And these are the bacteria that have to go out and collect them up and bring them to where they go. But they also create enzymes, and in fact, they are the factories. These little guys right here are what's called the factory that creates the enzymes. And what is the enzyme? The enzyme is one of the most elegant pieces of chemistry you've ever seen. Look at that. Proteins are enzymes. Enzymes are catalysts. Here it is right here. What's an enzyme? What's a protein? Well, these are the little beads that come in, and they turn into these enzymes and proteins, which are the products that go out and break down and kill invaders and do all that stuff. And they're created by using all of these 20 different amino acids. I'm getting a little deep right now, but I am going to get way deep into this. You're going to see all of this again, so why bother doing it right now? But what we want to prevent is this breakthrough right here of that layer, that specific layer. That's the fluid-filled highway. Once it breaks through there, then it nurtures itself off of your good living tissue underneath that has a lot of nutrients, and then it just starts growing and you become infected. And not just cancer, it's all invasive disease, chronic diseases. And this is the guy right here. This bacterial cell is supposed to create these ribosomes. which are programs. Remember, everybody's talking about RNA program? Well, that's what it is right there. Only they're making them in a lab saying, let's create this sequence, and we know it targets a certain thing. That's a shotgun approach. Your body, when it makes its mRNA programs, it knows how to make them, and they come out elegant, and they do the job correctly. Now, we're trying to do it quick, and on the fly, you don't make that thing quick. You can make it so that you know that specific type of enzyme has a specific effect on the body, whatever that specific effect is. But it doesn't mean necessarily it's going to fix the disease you're targeting for. Secondly, if you just make one little change here, you can cause damage. And it does appear that there may be some damage being caused. They're talking about the blood-brain barrier and some other things that nobody had ever thought about. And I've studied the blood-brain barrier very carefully because there's a guy down in Florida that just gives a shot of Interstitium right above, I think that's the name of it, right above the blood-brain barrier where these big globs can't get because it's so restricted. I think it's like 2 nanometers. It's just tiny. You can't, they come up there and they say, no, you can't get through, get away. So he goes up on the other side, squirts it in there, and minutes later, from a stroke, it seems to get rid of whatever that plaque buildup is, or there's another thing they call it, TNF, I can't remember. Anyway, I've seen the videos of it. They showed this one woman minutes later after getting the shot. She was back up and running just like brand new again. Very, very, very compelling looking evidence. So anyway, we're going to go into this deep. Stick around or do whatever you feel best about. But these colors are key. You see these different colors? Purple bacterium, green heliobacterium, these different colored. All of that color is critical. It's not just something they're throwing in there to make it look pretty. These are different. Chemistry makes different colors. Let's just go with that. And the different chemistry is different oxidative states, same materials, but they have different charges to them. The charge dictates the color. Blood is red or blue in us, basically red or blue-ish, which is oxygenated or deoxygenated. They show it, the 2 and 3 state, as green and yellow. But anyway, these are the different, because that's what it is in us. You have a 2 and a 3 state. The 3 state gives you the extra oxygen. Once you use that one up, it gets attached to somewhere else and gives off something else, and you end up with down to a 2 state. I'm going to get into this pretty deep. So again, you know, if you're interested, you're interested. And these are all the different types of bacteria and the different shapes, and that's what they can allow for a lot of different chemistry. Everything is about zones of magnetic influence. Basically, that everything is that. That's all there is, zones of magnetic influence. And they really literally create different colors depending upon the different magnetic attractions. So let's get started. If you're interested, stay with me. Okay, just to cut right to the chase. Bacteria versus enzymes versus chemicals. Here's the key. Bacteria make the enzymes, which are the chemicals. Simple as that. Bacteria are the factories that produce enzymes. Bacteria squirt enzymes out, and there are thousands and thousands and thousands and thousands of different bacteria in your body. And every one of them squirts out an enzyme, basically. They're the factories. These are the bacteria, single-cell bacteria. They float around your body using that propeller, that flagellum, to squirt around through this newly found layer, which is your interstitium. All right. This is just brand new stuff. Now we're going to start into some pretty serious stuff here, but remember this, the enzymes are not alive. They're just like the viruses. Viruses, they say they're not alive. Well, where'd they come from? They exist in these little capsules. They're capsids. They surround them, and they can go into the air, and they can float around, and then you get them in you, and it becomes part of your cellular activity. It's a program. Enzymes are the exact same thing, only they protect you. We see the enzymes as good things because they create chemistry very quickly in your body to break down food and attack things. They're good things. We see viruses as bad things. Well, viruses now, they're seeing viruses as good things, as good as most of them are good. The viruses, we always thought all viruses were bad. Well, so we created antivirals, and they may attack some viruses that are actually good for us. This is a very complicated web, and the key to take away from this right now is we have to determine what bacteria is supposed to be in your body and what quantities, and when a person is sick, what bacteria is missing. That's as simple as that. And the bacteria will have a color signature. With using artificial intelligence, we should be able to map somebody's bacteria in their body and find out what bacteria is excessive or is missing. And the only thing that creates enzymes is bacteria. They're the factories. So if you don't have the bacteria, you don't have the enzymes, you don't have the enzymes, you don't have the chemistry, you don't have the chemistry, you got problems. One way or the other, you're not going to be able to create something, you're not going to be able to break something down, you're not going to be able to attack invaders, you're not going to be able to create the chemistry that creates your body. You're going to be stiff, you're going to be sick, you're going to be sore, you're going to be open to invasion, you're going to be chronically ill. And that usually is tight junctions. Tight junctions are the rubbery, gooey stuff in between cellular membranes, and all of this stuff that touches each other has to be sealed. Otherwise, open up, right through. And then that's when you get this invasion right here, through this membrane. And they call it cancer, you call it anything you want, but there's all kinds of different diseases, and they are, basically all of them are a breakthrough of that layer. And that layer now, there's a video, well, I'll show you a quick shot of it, and then we're going to go into this. Again, this is going to be very long, so if you got a life to live, maybe. But there's only one way to do this, and it's just do it complete. I start right from the beginning. I went back to when they first discovered what SARS was, which is severe acute respiratory distress. It destroys your rubbery lungs. Why does it do that? Well, the rubbery stuff is collagens. There's 28 different types of collagens. Anyway, again, gonna get deep. Okay, this is one of the best videos I've ever seen about the immune system. Watch this. This is Go Call.
I still remember the moment. It's something I will never forget. The hair on my hands just stood up. It's a microscopic universe within each cell.
This is an unprecedented view of the cellular world, where we can actually see immune cells scooping up sugars in the ear of a zebrafish.
I just want to stop right now. I'm going to show you back at the interstitium. This is your immune system. That is that layer of interstitium. That is going to go out into the fluid-filled highway to go deliver whatever it's picking up here, which are probably enzymes or there's something. It's picking this up. They really don't know what they're looking at yet, but they have determined some things that are very important. These are the ones that gobble up the delivery stuff, and then there's other ones that are out sniffing around for invaders. Listen to what he says again. Let me just start back again. Here's Go Call, and he's from, I think, UC Berkeley.
The hair on my hands just stood up. It's a microscopic universe within each cell.
This is an unprecedented view of the cellular world, where we can actually see immune cells scooping up sugars in the ear of a zebrafish in real time.
Focusing only on the crawling immune cells, we've noticed two classes of them. One was not hungry at all, but it was very active in terms of trying to figure out, what the environment is.
You see this? This is basically what I'm talking about here. It's got a series of magnetic signatures here. And if it comes out squirting around here and it bumps into something that says, hey, you fit that magnetic signature, I got to go tell the guy that scoops up the stuff, come out and kill you. And that's what happens.
There's another one that was kind of slouching around with a lot of food in its belly. We can actually conceptualize and visualize and analyze the contents of each of these cellular compartments.
Let's watch this again.
Focusing only on the crawling immune cells, we've noticed two classes of them. One was not hungry at all, but it was very active in terms of trying to figure out, what the environment is. There was another one that was kind of slouching around with a lot of food in its belly. We can actually conceptualize and visualize.
All right, so these are inside of this immune cell layer, which is right here. That layer right there is the layer he's talking about. And it squirted that thing out and it went after wherever it had to go. And there was another one that's out here floating around in this stuff that's looking for invaders. That's how your immune system works, as far as I can determine. Okay, my friends, it just doesn't get any more exciting than this. Johns Hopkins revolutionary new gel cured 100% of mice with aggressive brain cancer. A new gel. Let's look into this because this is exactly what I've been calling for. Not necessarily a gel, but it's all the pieces are going to fit together. I'm going to show you uno momento. Okay, my friends, I'm going to tell you something right now. 2018, this changed every single thing about the human body. A newfound organ. Not just one little bit and piece right there. This covers your entire body. All your skin, your intestines, your stomach lining, your urinary system, digestive tract, 100% muscles, tendons, all covered with this particular layer, which is called the interstitium, which is a fluid-filled highway that sends all of these enzymes, bacteria, viruses, that's how they travel through your body. Now, and I'm going to explain to you what engraftment means in a moment, because there's a bunch of new articles just came out. Now, this is 2018. That's only 4 years ago that they even realized this existed. By the time they got into autopsies, it was flat, and they didn't know these fluid-filled pockets of bacteria were here, and that's what keeps you healthy. So, here's what I want you first to do. Right now, instantaneously, like spontaneous combustion, thumb up. All right? If you're unhappy at the end, thumb it down. Subscribe if you're not subscribed. Make sure you ring the bell so that you'll be notified on everything. Open your mind up. Now, we're going to end up needing funding and some help and some student researchers to do what I am about to propose, which is this bacterial analysis study I've been recommending for many years. And they just did it, and they were absolutely blown away. They did it with viruses. We need to do it with bacteria that is the product that creates the enzymes that makes your body run. Without enzymes, nothing will ever happen in your body. Zero. Not a single thing. Enzymes speed up the reaction so fast that your body can break down chemicals and put them back together and make you be able to do all the things that I'm doing right now, and do whatever you want to do. But if you don't have the right bacteria, you can't move. You can't get moved. Your gut gets bad. All kinds of problems crop up, and it breaks through this lining. This is where the bacteria lives, right here. And it fights with enzymes against anything that tries to invade. We're going to go deep. Get ready. Okay, there's a lot of talk about the viruses and about vaccines and RNA and DNA and all this stuff. Well, it gets a lot more complicated than that. But let's start off right from the scratch. What is the difference between viruses and bacteria? This is what they say. Bacteria are single-cell little creatures, basically, that can survive on their own. They're little zippity, they run around all over the place. They're inside or outside the body. They can live on you, but they're living, and they zip around with their flagellas and all that. Now, viruses, little different. They are enclosed in what they call a capsid. It's just a little film surrounding them, and they somehow get entering and multiplying inside the host healthy cells. So they go inside your cell and take over its mechanism and make it work to do whatever it tells us to do, because all it is a program. These are programs. That's all it is. Bacteria create programs. They create programs. They create programs. What is a program? It is an enzyme. What is an enzyme? It's a program. You want to see a program? I'll show you a program. We can't even make them as elegant as these programs. I'll show you a program, blow your mind. Okay, you won't believe how simple this is. These are bacteria, and there are thousands and thousands and thousands of different species. They just cataloged 30,000 new viral species, which is not much different than this. These are bacteria, one step different. These are, well, we'll get into that. But inside of these bacteria, there is what somebody's called ribosome. You see that little blue dot? Well, what is a ribosome? It is literally a program. There's another program in here, which is the DNA. This DNA says, I am a certain type of bacteria. I am going to make an exact ribosome. And what is a ribosome? A ribosome is an enzyme. And what is an enzyme? An enzyme is a protein that goes out and does havoc on other molecules. And how does that thing come out of here? It squirts it out in a little bead just like this. And once it's all squirted out in the exact sequence of colors like that, it goes and it turns into like a protein, which is like this. And it will be exactly identical every single time. Look at this. Look at that. What happens is that there's about, I don't know how many there is of these things, but they're all magnetic, and there's 20 different amino acids. And once they get out, squirted out to the full length of whatever it is, it just goes, and there it is. Now, it'll be exactly this configuration with all these little polarities sticking off the edges. See these little polarities? Those are the things that it's a magnetic signature. That gives it click chemistry. What is click chemistry? Click chemistry means this is magnetic. It's a big magnetic block. As it travels through the body, if it finds a magnetic signature that matches its magnetic signature, it attacks and goes click. And every one of the ones that it clicks up against, which is usually in a mass, which is a tumor, they all die instantly. Just like instant. Snap. And all of the magnetic ripple goes through, and these become denatured. They no longer can do their job. But guess what? The guy that attacked them, the enzyme, the protein that we made, it just keeps going on its way and click, click, click until it gets all the ones it can. Or it dies or whatever happens to it. But then eventually it just goes away when it's not needed anymore. But one single enzyme can wipe out hundreds of thousands, millions of attacking enzymes, basically viruses, whatever they are. This clicks it and they just die. That's what immunity is. But it has to get into your system in that layer that I just showed you, which is this one right here. It's got to get in that system right there. And it comes from that layer, which is your immune cells. And that's where your bacteria resides. And if it needs something to attack something right here, squirts it right out and it goes and kills it. It may have to come from way over here. I don't know. But what happened was they found out this gel they were putting on these rats to protect them from brain cancer wouldn't work unless they had surgery first. Well, it worked, but not very well. It worked 100% of the time when they had surgery first. Well, why? Because they removed the tumor? No, I'll tell you why. All right, see this? While analyzing the genomes of single-celled microbes, those are basically bacteria, a team of researchers made a startling discovery. Thousands of previously unknown viruses were hidden within the microbe's DNA. Well, are those actually viruses, or are those oh boy, are those, wait a minute, ribosomes. I know, it just recalled. Here we go. What happens is inside these bacterial cells, which is what they're talking about, bacteria, they create a package called basically a ribosome. And it's a chain of polypeptides. And there's amino acids that come out, and then they get programmed to do a certain job. That's what a ribosome is. And which is, I believe, a virus. I don't see any difference whatsoever. None. Zero. Between a virus and a ribosome. And this is where the ribosomes come from. They come right out of the single-celled bacteria that they were talking about. You don't lose focus because of all these little words they throw in. Microbes are simply nothing more than bacteria and those little tiny things. They're organisms too small to be seen without using a microscope. Bacteria, archaea, single-cell eukaryotes, all kinds of little critters that are inside of you. I've been saying this for years and years and years. They say that they found more than 30,000 viruses built into the genomes of single-celled microbes. I just showed what a microbe is, bacteria, all that stuff. Now, they explain that viral, which is a virus, DNA, might enable a host cell to replicate complete functional viruses. Well, that's just to make more of themselves. We were very surprised by how many viruses we found through this analysis. Keep calling them viruses. I call them enzymes, says the lead author, Christopher Bellas, an ecologist who studies viruses at the University of Innsbruck in Australia. In some cases, up to 10% of a microbe's DNA turned out to consist of hidden viruses. That's what they're made for. They're made to create these little chemistry packages. They call them viruses. I'm calling them ribosomes, basically. They're the same thing as ribosomes. I don't know why. Anyway, these viruses, which I'm calling enzymes, don't seem to sicken their hosts. The researchers say, and they might be beneficial, which is what enzymes do from beneficial bacteria. It's called probiotics. Some of the new viruses resemble virophages, which kill things. A type of virus that infects other pathogenic, which means bad guy, viruses, attempting to infect you, who is the host. So why so many viruses are found in the genomes of microbes is not yet clear. Our strongest hypothesis is that they protect the cell from infection by dangerous viruses. Yes, they're finally admitting to this. This is incredible. There is so much, every one of these viruses they're calling is an enzyme. What is an enzyme? It's a chemistry package that does a job. I'll show you what an enzyme is. Hold on. All right, if you want the deep details, you can come up and look around. But what does an enzyme do? In a puzzle, there's only two pieces that fit together properly. That's what an enzyme does. An enzyme will do one specific job, and it's called a catalyst. It catalyzes stuff. How does a catalyst work? A catalyst does chemistry breakdowns at the speed of light, literally. One enzyme molecule can change hundreds of thousands of other molecules that are the ones that is the other piece of the puzzle, just like that. Hundreds of thousands to one, and then that one goes on to go and break another 100,000 or more. All right, so we know an enzyme will only work with one specific molecule, and they are so elegant, these enzymes, that the molecules are elegant. This is an enzyme right here. That's an enzyme. It's called a protein, an enzyme, a catalyst. All of those little particles have to have somebody that they can talk to. And when they all meet together, the whole thing pops and it's done. Now, if you don't have that enzyme, you're going to be sick. That makes chemistry. And without that enzyme, you can't attack things that are trying to attack you. You can't break down food that you need to break down into its component parts so that you can absorb them. You can't destroy toxins. You can't do any of the chemistry your body needs to stop this. This is cancer. And cancer is just another form of invasion. You have all kinds of invasions in your body. Cancer is one of the ones that is it's more invasive than other types, but everything breaks through that particular layer. That layer, remember, was that interstitium. Once it breaks through there, you're done. Then it has a source of stuff to grow on, and it starts to feed off your body, and it just keeps getting bigger and bigger and bigger. Now, what you have to do is protect it before it gets through these membranes. There's all kinds of layers of membranes, and then you get to that layer. Now, what they never realized was the fluid-filled highway is right above this layer, and they still really don't. I don't think anybody pays attention to it, but here it is right here. That's the fluid-filled highway. This right here is the layer. You see, that's the layer, and they have seen the bacteria living in this layer, scooting out into the fluid-filled highway to go deliver their package. And they have seen the two types of bacteria. One is the sniffer bacteria that goes out and looks for the bad guys, and then the other one goes in here, scoops up all the stuff to go out and deliver it. And University of Caltech, I think it is, a guy named Gokul, nice guy. He shows it. He shows this happening in the new photography they can do. As of about 2018, I think they started to realize they could see inside of here. But this is the interstitium, and that is the layer of immune cells, and this is the fluid-filled highway. And through that fluid-filled highway, these bacteria send out their packages. Now, I talked about engraftment before. Engraftment means you need to get these bacteria living in here. They have to breach the defenses of here to get in here. Once they're gone, they're gone. They don't want to ever come back. Nobody wants them ever to come back again because they have all of these things that say, no, you stay out. I don't want you. If you can break through and get into there, then you're okay. That's why 100% when they had surgery, this is all cut open. And then that stuff engrafted into these fluid-filled highways and started living amongst where it's supposed to be. And then there was 100% cure rate, 100% across the board, only with surgery. Now, what that tells me is if we knew what the bacteria was, you could literally cut yourself here and squirt it right into there. That, as far as I'm concerned, once it's in that fluid-filled highway, you're all right. But until you can get it in there, it won't get engrafted. It won't live amongst its buddies that are supposed to be living in this immune layer. All right, so this is critical. The engraftment's critical, the type of bacteria, the enzyme, and we need to do a study. They just found 30,000 viruses. We should be able to find all the all the bacteria that's living in your body, and now with these new AI programs, every bacteria will have a signature. You saw what a bacteria looks like, or well, actually you saw a protein enzyme, but it's still going to have a color. You see all these different colors? They're not there for nothing. They're because there is different energy levels. When there's a different energy level, there's a different color. Now, this is a basic about what are enzymes and proteins. That's what they are right here, and they are powerful and and you have to have them. Now, these transition metals, you see all this? This is what carries all the stuff through your body, all these different positive states of oxidation. They can pick up stuff and dump stuff, and they have to carry stuff around, but they have to have the stuff broken down to be able to attach to these various oxidation states. And if you can't break it down, you're done. And how does it get broken down? Well, enzymes. As I showed you, they can, just like that, wipe out hundreds of thousands of them at the same time. And this is how they get programmed. Remember I told you polypeptide chains? These are the same thing here. And some bacteria squirt out 2 or 3, some squirt out 50 or 60, but whatever happens, at the end they all come through here and they get programmed with this transfer RNA. There's 20 different amino acids. That gives you a program that's just stunning. We have ones and zeros. We got 20 different codes here. And that's why you see all these different colors. And the range of programming that comes out of a single strand of these codes, it's beyond anybody's possible way to understand. It's just way beyond any program or anything. I don't think anybody could. Well, actually, apparently they are finding these codes. Some of this new equipment that they can analyze chemistry with is pretty impressive. All right, I've said that bacteria, your viral infections and bacterial infections are basically the same thing, and they say the same thing. It says, in some cases, it's difficult to determine whether an illness is viral or bacterial, because many ailments, including pneumonia, meningitis, and diarrhea, can be caused by either one. So they both, you have to have a source to create that capsid, which is the little capsule that has that program in it. Once that program gets in your body, it's not like alive, but it's going to find an entrance into one of your cells, and then it's going to take over that, and then it starts to manifest itself. And then they become a growing mass, basically. All right, I don't remember if I showed you this yet, but Johns Hopkins revolutionary new gel cured 100%. All right, of these mice with aggressive brain cancer, but only if they had had surgery. That's the key. Now, researchers at Johns Hopkins have developed a novel hydrogel. Now, listen to this. Hydrogel just means water. Hydro is hydrogen and gel. Hydro is water. That has successfully cured 100% of mice. 100% with aggressive brain cancer, specifically glioblastoma, which is the deadliest. The hydrogel combines an anti-cancer drug and an antibody. The antibody is the key, and is applied to the brain after surgical removal of a tumor. It only kills them if it's after the surgical. If it's not after the surgical, it's only 50%. Hold on one second. See, here it is right here. Surgery is essential, the researchers said. Applying the gel directly without surgical removal of the tumor resulted in 50% survival rate. Well, I bet if they took half of the tumor out and left that gap there and flooded it with this stuff, this gel, it would suppress that tumor. I'd be willing to bet. I'd like to see them try that. All right, they have a YouTube about this. I think maybe I'll take a look at it, but it's self-assembling hydrogels. As soon as you put them in the water, they assemble all by themselves. Video clip shows how the liquid substance self-assembles into a gel when injected into a saline solution, and we are saline creatures. Now, this is Johns Hopkins University. By the way, I went there to take genomics with Professor Steven Salzberg, and the gel solution consists of nano-sized filaments. That's molecular size, atomic size, made with paclitaxel, an FDA-approved drug for breast, lung, and other cancers. Well, I'm not sure about how this works. It's the first I've seen of it, but what they end up talking about is the filaments provide a vehicle to deliver an antibody called a CD47. An antibody. Antibodies are very, they're molecular, a big chunk of molecule, which is an antibody. It's called a CD47. By blanketing the tumor cavity evenly, the gel releases medication steadily over several weeks, and its active ingredients remain close to the injection site. Well, that could very well be. I'm thinking that it's become embedded into that layer that I've been talking about, which is the immune system right here. It's engrafted. So now it lives in there, and no longer will it allow these things to come in. Something has killed off bacteria in a lot of people in a lot of ways. I mean, there's toxins in all kinds of stuff, and a lot of it is lymph node stuff, where your lymph is the dump. they bring all the toxins to the lymph nodes and say, clean it up and get it out of here. And if the lymph nodes don't have the bacteria to break everything down, it breaks through. And that's what happens right here. You get this kind of an invasion. Lymph nodes. And that's why they say, oh, it's metastatic, it goes all over the body. No, you don't have the enzyme anywhere in the body. No, it's just going to take one at a time, and it's just going to keep going. That's the problem with this. If you don't have the right bacteria to fight back, it's one of them's going to go, then another one, then another one. Now, how do we know what to do? We need to do some studies. And we need to be able to do just what they did with these other viruses. 30,000 of them they found. Watch this. See, I probably showed you this before, but I want to make sure you understand. Science alert. Scientists have found 30,000 new viruses in DNA microbes. They never looked before. They're totally stunned. This just came out. While analyzing the genomes of single-celled microbes. Genomes are nothing more than the sequence of these little beads, basically. They're the genetic signature. And they have to be in a certain order to work correctly. If one of them goes wrong in a certain way, you're done. So they were analyzing these sequences of single-celled microbes, which are these little, basically bacteria. A team of researchers made a startling discovery. Thousands of previously unknown viruses were hidden within the microbe's DNA. Well, let's just take a quick look at that. Where were they hiding? All right, and where is the microbes they're talking about? Let's just take this for instance. These are the ribosomes, and that's where all these things were hiding. Now, they say maybe they were hiding in these DNA loops. I don't know. But ribosomes are the packages that are created by bacteria. That's a bacteria, single cell, one cell. It has its own program built into it, which is the nucleoid DNA. That's its own program, and its program says, we make this little package here, ribosome. And what is that package? It's exactly that. If it's not exactly that, it didn't work. That means you have some kind of defective problem. But normally when it squirts it out, all these magnetic signatures are going to hook together, and you got exactly what you wanted, which is your enzyme. That enzyme does the work in your body. Otherwise, that work doesn't get done. And here's what it looks like at the end. And that can change molecules, hundreds of thousands of them, within 1 second using click chemistry, as I believe I have explained. And then they go through this fluid-filled highway to go deliver those products wherever they need to deliver them. It goes right through here. All right, you see this? These are the things that carry around things in your body. They're called ligands, which are the little pinchers, transition metals. This is what's in your blood. Your blood is saturated with these metals. And here's what it says right here. They're called ligands. Now, transition metals are all these different titanium and copper and all that, form colored compounds and complexes. These colors can vary depending upon the charge on the metal ion and the number and type of groups of atoms called ligands, which is a pinched part, which is attached to the metal ion core. In aqueous solutions, that means in water, the ions form complexes with the colors shown to the right. You see all these different colors. There's iron. They're showing green and yellow, but I see red and black, but I don't know. That's when it becomes oxidized. Now, are these within the blood? I don't really know more than to tell you that these are the different colors you're going to find in a lot of mud fossils. And especially in opals. I mean, opals are just exactly like this. They have all those transition metals. Opals were preserved in a certain type of chemistry. I'm not exactly sure what it was, but it was saturated with blood, which is the transition metals. This is an eyeball. That's actually the muscle strap. And this is the colors that it took on. Now, here's a heart. This is literally a heart. And all of this is that, like, from the ventricle walls, want it to be blue. There's where the color of the blood is, obviously. It was laying this way, and the metals all collected here, because they're filled in all your blood. And all of these are little different tissue areas of different colors that wanted to attach to these things. This is what creates opals. They're in living tissue. They migrate, like this is a clamshell. And they want to find a metal that will attach to them and stay there. Normally they come and go. In your body, they move through your body and they go back over to the liver and get cleaned up and all of that business. But once you die, it stops moving. And just the water flowing around and so forth with all this chemistry in it, sooner or later, if they hit the right places at the right time, they lock in and they become that color. That's what opals are all about. All right. Most people don't think a whole lot about their heart beating and their blood flowing through their system. As long as you're walking around and you're breathing oxygen and you're feeling good, well, you're good to go. Well, did you know about the blood-brain barrier? Most people probably don't. But if you do, there's a little more to it than just it's a barrier. How big is it? What does it let through? What does it keep back? As I showed you, I believe I did show you that they put that gel right directly on the brain. Now, that gel, if it had a very sophisticated chemical like this, which is a protein enzyme, it may not be able to get through there. There's a doctor in Florida treating people by giving a shot above the blood-brain barrier, and they recover from stroke. It seems to be, some of them do. I'm not going to say everybody does. But this is the deal with the blood-brain barrier. Brain diseases can be difficult to treat through a systemic delivery. That means through your whole body. You can't put it in your arm because it's not going to work. Delivery of therapeutic agents, because most agents, that's a big statement, most are blocked by a natural barrier in the brain called the blood-brain barrier. And it is tiny. It's like 2.3 nanometers or something. I mean, it's just tiny. It's so small that it will only allow certain molecules up into your brain. Now, they're trying to open it up by focusing ultrasound, and I guess they did, but that doesn't seem, I don't know. Anyway,